Welcome back to my channel Back and By World. In today's video, I'll be discussing the retrovirus, that is HIV, and I'll be discussing what are retroviruses and the structural components of this virus. Okay. The first question here is, what are retroviruses? Retroviruses, these are viruses which are having RNA as genome. Okay, it is having RNA as a genome and RNA as a genome, and it replicates in a host cell by making a RNA DNA intermediate. Okay, when it infects the host, okay, it infects the host, it replicates, its replication in the host cell is through an RNA DNA intermediate. Okay, here the RNA DNA intermediate has started to form, it has completed in this step. The RNA DNA intermediate, it is not stable, and the RNA intermediate is usually degraded and double stranded DNA is synthesized, which goes and integrates this double stranded DNA. It goes and integrates into the host cell nucleus, into the DNA of the host cell. Okay, let's come back to the definition. Retroviruses are viruses having RNA as genome that replicates in a host cell by making RNA DNA intermediate, then into double stranded DNA. The DNA molecule gets integrated into the host cell DNA, where it replicates. Replication takes place when the cell replicates its own genome. Okay, that was it about it. Now the structural component of the one of the retroviruses, that is HIV, that is human immunodeficiency virus. Okay, here I'll be discussing the structural component, that is what are the components of the HIV virus. Okay, so there are a number of components. It includes surface proteins, transmembrane protein, reverse transcriptase, integrase, capsid protein, matrix protein, proteases, nucleic acid binding protein, and a link protein. Okay, where the GP here stands for glycoprotein and P stands for the protein. Okay, let's begin with the components. The surface proteins, they are two in number, that is GP160 and GP120. These are the glycoproteins, and transmembrane protein is GP41. Okay, the reverse transcriptase is the enzyme which is usually found in the HIV, and it is responsible for the synthesis of the those RNA DNA intermediates and then double stranded DNA. Okay, the enzyme which is responsible for its replication, it is reverse transcriptase, and it is protein 66, protein 55, and protein 51. Okay, next integrase is another enzyme, and we saw in the last slide that after the formation of the double stranded DNA, it gets integrated into the host cell genome. Okay, the enzyme which is responsible for the integration of that double stranded DNA into the host cell genome is the integrase, that is P32, protein 32. Okay, next is the capsid protein P24, matrix protein P17, and a protease, that is P12, and nucleic acid binding protein P7, and a link protein, that is P6. Okay, here on the left hand side, this is a very general, we can see a diagram of a retrovirus. It is usually having a single stranded RNA as a genome. Okay, and usually which is usually which is enclosed in the capsid. This genome is enclosed with the capsid, which will be having the capsid proteins, and it will be having an enzyme, reverse transcriptase, and an integrase as well. Okay, and then the capsid is surrounded by an envelope, okay, which is having a number of we can say the surface proteins and the transmembrane proteins, which usually responsible for the we can say entry into the host cell. Okay, the proteins responsible for the entry into the host cell, that is in the are present in the envelope, and in case of the HIV, it is GP140, that is and GP41. Okay, that was all about today's video. Thank you so much for watching my channel. Please like, subscribe, and share.